Hi, I'm Dr. Mohammed Idris, Senior Principal Scientist at CCMB. My research focuses on understanding the complexity of organ and tissue regeneration in alternate animal models such as zebrafish, marine ascidians, and echinoderms. Also, we are trying to understand the biomechanism of degeneration of neuronal structures due to genetic, chemicals, stress, and aging. Amputation of human limb will never lead to any regrowth or regeneration. The maximum happens is wound healing if given a treatment. Whereas in zebrafish, which is similar to us at physiological, anatomical, and with 70% genome similarity, can regenerate the entire caudal fin tissue, which is similar to human limb, within 7 to 10 days post amputation, maintaining its architecture, and more importantly, it terminates the regrowth at the end of regeneration. Understanding how zebrafish regenerate their fins, which is similar to human appendages, can provide insights into promoting similar regenerative process in humans and also to understand the cancer in a much better way, as regeneration and cancer share same genetic mechanism and cellular process.
We use high-throughput genomics like transcriptomics involving next-generation sequencing and quantitative proteomics involving labeled approaches to identify and associate various genes and proteins involved in regeneration and in degeneration.
Based on our recent molecular studies, we have found nearly 1,400 genes and 600 proteins that are closely associated with regeneration through differential regulation at various time points of regeneration post-amputation in zebrafish as well as starfish.
Regeneration is not merely a regrowth, it terminates upon completion. It can potentially lead to a better understanding of complex diseases and therapies.